So because we're not going to be able to meet together and do this lab as a class, we still want you to understand the theory behind it because this is a really important biotech procedure that is used especially when you need a quick test to verify the presence of a protein. So the simulation we were going to do, ironically, was tracking a disease outbreak where one person was going to be infected and we were going to go through a couple of rounds of them sharing fluids and then see how rapidly the disease will spread. Now, we were going to use just a chicken protein to simulate this, not an actual virus, obviously, but this was to help you understand this biotech procedure called an ELISA. ELISA stands for Enzyme-Linked Immunosorbent Assay. Now, ELISAs are powerful, and the reason for that is because they're tests that can be done very specifically on any protein that you want to look for, and in some cases, these tests can take 5 minutes. For example, a pregnancy test is an ELISA. You pee on it, the presence of a protein, a hormone that is generally found in urine after a certain number of days of a woman being pregnant show up. They go through this process and they show up positive or negative. That's essentially what an ELISA is. Pregnancy test is one of the most common types of tests done today. So these tests, however, have their drawbacks because they're extremely specific. You have to know which protein you're looking for. That's one of the reasons why we did not use this test for the GMO lab. The GMO lab used PCR, and PCR was used to essentially look for a wide range of genetically modified plants with some commonalities between them, right? The CAM-B promoter and the NOS terminator. But if we wanted to look very specifically at just one GMO, we would have to know what protein was being made, and we'd have to actually have the test designed to look for that protein, but it wouldn't work for anything else. Now, why are pregnancy tests very commonly used? Because the protein is the same in all women, and people are always wondering if they're pregnant or not. And so this is something that has a high rate of return to develop this test against that protein and see if you're pregnant or not. Now, so that's why a lot of these tests are developed. You have to kind of have a reason to develop them because they take a while to actually generate these tests. A lot of money, too, put into creating that. Once you do, then you can actually get a high rate of return, and these are very rapid. Like I said, minutes. You can do these in minutes, which is why we were going to do this in our class, is because it would have taken just a couple of minutes to determine whether you spread the disease or whether you're free and clear after so many rounds, right? So there are certain real-world diagnostics. Some GMOs are used for an ELISA, drug and steroid testing, especially if we know the chemical that we're looking for. Pregnancy tests, biodefense, mad cow's disease, SARS, HIV. These are kind of the most common diagnostic tests being used today because they're so prevalent, right? We're always testing for drugs. We're always testing for steroid use. We're testing for pregnancy. We're testing for certain diseases. You got to have some really fast testing for some of these things that are really dangerous to make sure that you keep ahead of it. And that's one of the difficult things, especially today with this outbreak, is we use PCR to determine whether or not people are positive rather than an ELISA. Now, let me explain how an ELISA essentially is made. And you'll begin to see why we haven't quite used it yet and ultimately how hard it is to generate some of the reagents for this test, as quick as it is. The way in which you generate the preliminary reagents for this is you have to take the protein that you want to detect and you put it into an animal, and the animal will actually create an antibody against that protein. And that antibody that they create is used in the ELISA. And so when first creating the pregnancy test, they isolated the protein that women, the hormone that they make when they're pregnant, and then they put it into an animal to create these primary antibodies that would actually attach to it. And that's a very costly process to be able to create those primary antibodies. So The antibodies are very specific for the protein shape. And that's one of the powerful reasons of using these in the ELISA is they're able to only bind to and attach to that protein and no other protein so that you're not getting false negatives or false positives, right? You don't want to detect something that's not that protein and say, oh, you have the virus and you really don't. Or you want it specific enough so that it's not, you know, if it doesn't attach to the right thing and say, oh, you're good, but you're actually positive, you don't want those false negatives, right? So you got to make sure that this antibody is and will bind to that protein that you're interested in. And that takes lots of money to be able to generate that antibody structure. So this is basically how an ELISA works. You put the sample, be it the blood or the fluid that you're looking for, or the pee. If you're doing a pregnancy test, you pee on the stick and it runs through this process. So this is done in these little things. I'll show you later on how basically a pregnancy test is able to go through the same process as you can see right here. We'll talk about how it's done. It's the same way. It's just done on a stick rather than in a little container. So the first step is you take your sample and you put it into the little well, and any proteins that are in that sample will actually bind to the bottom of the plastic, okay? Now, before you go into this next step, and this is a really key point, especially for the exam, I'm going to test you on this on exam 4, where before you do the next stage of this ELISA, you have to actually coat the remaining surface of the plastic with some other protein. Generally, we use like a milk protein or a detergent protein or a chemical or whatnot to make sure that any space that hasn't had the protein bound to it gets something else attached to it. And the reason for that is because if you don't do that between this step and that step, when you put these primary antibodies into the next step, if there's any space left on the plastic, these primary antibodies will actually bind to that indiscriminately. What will you end up with? You'll end up with a false positive, right? In some cases, if you don't do that with the negative controls and the antibody binds to that without any antigen, then you get someone showing up with the positive result and you're like, oh yeah, you've got the virus, when they don't. And that can change a number of different things. And so whether it is the actual protein or if there's nothing in there at all, you have to make sure that between this step and the next step that you actually have this detergent that comes in and basically fills any of the space, right? For your negative controls, you typically don't have anything in there. And so you have to make sure that that detergent binds to the bottom of the wells. Otherwise, when you put the primary antibodies in there, they'll actually attach to the bottom and it will show up as a positive result instead of a negative. So that's why you have these negative controls. You got to make sure that between the first and the second step, you coat the bottom of the plastic, any space that's available, whether it's the positive or negative, with that detergent to make sure that none of these primary antibodies bind to any other surface except for the protein that you're interested in, because they themselves are proteins. That's what antibodies are. But these antibodies will specifically bind to that protein that you're interested in. Now, the third step is you actually attach another antibody to the first antibody. These are what we call secondary antibodies. Now, what's so special about the secondary antibodies? These secondary antibodies actually have an enzyme attached to them. That's where the name of this assay comes from. Enzyme-linked immunosorbent assay. Okay, so that secondary antibody has an enzyme attached to it. Now, these secondary antibodies are designed to attach to the primary antibody. So it almost creates this sandwich where on the first layer you have the protein. The second layer, the primary antibody binds only to that, and then you wash out anything that doesn't have the primary antibody attached to it. And then the secondary antibody comes in and attaches to the primary antibody. Now, what happens with this sample is that you use a certain chemical that in the presence of that enzyme, the enzyme will actually metabolize it, and it starts off as a clear chemical, but as the enzyme metabolizes it, it turns blue. And so anytime you have a positive result, you're going to see the color turn blue. And that's because you have the antigen, which is the protein, the primary antibody attaches to that, and the secondary antibody attaches to that, and essentially you get this blue color. So that's what the positive control is to ensure that the assay actually works. So we use a very specific known protein here that we know will show up. Now the negative control, this is to make sure that you don't get false positives. In these samples, you don't put any of the protein, any of the antigen. You coat the wells afterwards with that detergent, and then when you put the primary antibody in there, it has nothing to attach to. So when you wash these cells out, then the primary antibody washes out. Then you put the secondary antibody in there, it has nothing to attach to, so it gets washed out in the third wash step or whatnot, and there's nothing in there. Then when you put that clear chemical in those wells, there's no enzyme there to metabolize it, so it stays clear. So that's why this is an indication that there is no antigen there. There is no protein that the primary and the secondary antibody is bound to. So that's the negative control. Then you have your samples, and we do them in triplicate just to make sure that they're being done right. And these samples are basically what you get from when you are sharing the fluids with each other. And that's what we were going to simulate with the outbreak, was one person was going to have it, nobody else was going to have it. That one person would have spread it to another person, and then they would have spread it to another person, and then they would have spread it to another person. And by the time we got done, over half the class was essentially going to be infected. And we were going to try to trace the outbreak back to the original source. Now, the only way you could have done that was essentially to keep track of where you've been, who you've been with. And that's why in the outbreak scenarios, when they find someone who's positive, they're like, okay, where have you been? Oh, I was in Europe. Okay, where were you before that? Oh, I was in, I'd come from the UK and whatnot. Okay, where were you before that? So they follow your steps, and then they start going to other people and testing them. And they're like, oh, they're negative. Okay, you didn't get it from them. Or they test someone else and they're like, oh, you're positive. Where did you go? And so that's essentially how you're able to try to track the origin of these. Now, this has to be done in real time. Otherwise, you're never going to be able to keep up with it. And that's one of the difficult aspects of doing PCR, because it's so slow. By the time you get the results, if people haven't self-quarantined, they'll have already spread it to other people. And so this is one of the simulations that we were going to essentially do. So for this class, unfortunately, we're not going to do a write-up, but I am going to give you a small quiz that's going to test you on some of the theory that I talked about in this lecture, especially when it comes to how the process is done. And part of that is because on exam 4, you're also going to have some questions on this process. So let's kind of go through how the pregnancy test works to help illustrate that idea. Now, the pregnancy test is a little different than how we're doing the ELISA. Basically, these antibodies right here already have a pink color to them. And when you're looking at the stick here, if you were to actually look at this part of the stick, you would see a high concentration of these pink antibodies that are specifically designed to attach to the protein. And what happens when you pee on this stick and the proteins start migrating through capillary action up through the stick, as you can see right here, is once these proteins attach to these antibodies are going to start migrating up this way. Now, this first strip are also the primary antibodies. These are fixed in place, and what happens is when these antibodies that have the attached protein to them also attach to here, that's what creates that first line. That's what tells you that you're positive. But you also have to make sure that you have a control here. And so that for whatever reason, you didn't just get these stopping at this point. You also have to make sure that this is the verification that you actually had the process working. And this is where any leftover antibodies that haven't bound to here, they're going to hit these secondary antibodies right here. And these are going to attach to the primary antibodies and basically catch anything that went by, thus saying, okay, it wicked all the way up through. Again, if there were no proteins present, all of these primary antibodies would have passed by this region and just stuck to this part right here, right? So they only bind to these primary antibodies right here if that protein is present, as you can see in this part right here. The secondary antibodies are over here, they bind to here, and that's just a control to make sure that the assay worked as it should. Okay, so going back to that sandwich area, this is just all of the procedures and protocols that we would have walked you through, and obviously you're not going to do the steps of the assay, but fundamentally want to make sure that you know how this was done, and that's essentially what you're looking at right here. Is the first step is any protein that's in your blood that they take from a cheek swab or a blood sample. They're going to isolate the protein and they're going to put it into this and kind of see whether or not that, if it sticks to there. And then they're going to put the primary antibody in, which is going to bind to that protein. Then they'll put the secondary antibody in, which has that enzyme. And then they'll put the coloring reagent in to get it to change that color. Again, if there is no protein at the bottom of the well, the primary antibodies have nothing to stick to. So the secondary antibodies have nothing to stick to. And so the color reagent's not going to be affected because between each step, you wash out anything that's not bound to the bottom of the plastic. So you got to form this little sandwich of protein, primary antibody, and secondary antibody if you're going to get any type of color reaction. So that's why I told you to make sure you understand that if you don't treat both your controls, your positive controls, and your negative controls with a detergent, and you do that for your samples as well, then you would actually get false positives. Your negative controls would show up positive because even if there was no protein there and you didn't treat this with that detergent, the primary antibodies would bind to the plastic, then the secondary antibodies would bind to that, and then you would get a color change when you put that clear thing in there. And so that's why you have to make sure that that's coded between the first and the second, and that's going to show up on your exam. Okay, so that's the ELISA. Again, what's the difference between an ELISA and a PCR? ELISA looks for the protein. PCR looks for the DNA. So with the COVID-19, they're using PCR to look for the presence of the RNA sequence that's specific for that virus. One of the difficulties of testing for COVID-19 with an ELISA is some of the proteins on the surface of that virus are the same proteins on other coronaviruses, that same class of virus. So they'd have to find a protein that is unique to that virus to be able to create an ELISA that is specific. They'd have to go through a number of tests just to make sure that other viruses do not show up, thus creating a false positive from that test. So that's one of the reasons why they haven't developed an ELISA for it yet, because it takes a while to get that very specific, quick, inexpensive, low-tech. You can do it in a matter of minutes, and that would be the preferred method. But like I said, a lot of prep that goes into that. PCR, this is one of those that is expensive, but can be specific for that virus without a lot of prep work. And so this is why some countries were able to get these tests out very, very quickly and be able to start testing people for it right away. Okay, so that is the ELISA. I'm going to test you on some of the theory behind how it's done. I'm going to give you a quick quiz just to make sure you're up to speed on that theory so that you're ready for the exam number 4. but I'm also going to generate a lecture that is going to review for the exam and make sure you know everything that you need to prepare for that exam.